You're listening to Health Professional Radio. My name is Wayne Bucklar, and my guest today is Geoff Cumming. Geoff is the CEO of Anteo Technologies and joins me from Australia. Geoff, welcome to Health Professional Radio.
Wayne, it's a pleasure to be here.
Now, Geoff, I have to say, Anteo Technologies is not a name that immediately tells me what it is you do. Can you fill me in on what it is that you do?
Yes. Anteo Technologies is a subsidiary company of an ASX-listed company called Anteo Diagnostics Limited. Anteo Diagnostics Limited has a technology that is basically a nano glue, a glue that operates on the nano scale. The inventor of the technology is still with the company, a fellow by the name of Joe Meiji, and the company was founded early in the 2000s. Initially, the technology is being used in a healthcare environment and specifically in pathology tests, and even more specifically, immunoassays. Generally, with immunoassays, you try and bind an antibody to a solid phase. People have been doing this for decades now, but conventionally, either passive binding process is used or a covalent binding process. We use coordination chemistry, and as a result, we tend to be a little more gentle on the antibodies. So conventionally, using covalent chemistry, for instance, you might get 1 out of 5 antibodies that remains functional when bound to a solid surface in the creation of an immunoassay. With our process, 5 out of 5 remain functional.
So your nano glue increases the effectiveness of the analysis process? Is that the bottom line with it?
That's the bottom line. Because we've got 5 out of 5 antibodies versus 1 out of 5 functional, what happens is that we can either increase the sensitivity of the immunoassay if there's a clinical need for the sensitivity to be increased. Most of the immunoassays that are available on the market at the moment have got all of the clinical sensitivity that's needed. If that's the case, we can produce an immunoassay that has the same level of sensitivity as the existing test using as much as 90% less antibody. So it's either a better test or a less expensive test. But perhaps more importantly, there is a trend in the industry towards miniaturization. And conventionally, in the Australian context, you'd have Sonic or other large pathology labs doing these immunoassays, and they've got large bench-top or even standalone analyzers that crunch through thousands, perhaps even hundreds of thousands of tests per day.
Mm-hmm.
There's an increasing trend towards miniaturization so that in an ambulance or in emergency departments or at bedside, doctor's office, or even at home, you can perform these immunoassays. Well, with increasing miniaturization, it becomes increasingly important for each and every one of those binding interactions to be effective. And so we find ourselves ideally suited for that trend towards miniaturization because all of our antibodies retain their functionality, whereas that's generally not the case with either passive or covalent coupling.
Geoff, it sounds like a technology that I should have heard of as an Australian product, and I confess I haven't. But thank you for sharing that with us, because it is an explanation that makes perfect sense. I've read your website and read some of the technical spec, and nano glue is exactly the description I needed for my non-clinical head.
Very good. Well, because it is a nano glue, the background of virtually everybody within the company is healthcare. So our first commercial foray, has been into the healthcare space, specifically immunoassays, but there are broader applications for that technology within healthcare, such as bioseparations, where you're trying to separate, let's say, antibodies from plasma or blood to purify antibodies, or even in medical devices, where you want to bind things to one another, and it's not always easy. What we say is that we can give you the surface that you want with the surface that you have. So we have an almost infinite ability to transform hydrophobic surfaces into hydrophilic surfaces, or hydrophilic surfaces into hydrophobic surfaces, and everything in between. So that means that depending on what the need is for immunoassays or bioseparations or medical devices, we can provide what's best suited to the particular application. But again, because it's a nano glue and the need for glues is ubiquitous, we've got capabilities outside of healthcare as well. And in fact, earlier this year, we filed a patent that applies our technology to the field of batteries.
I see. So it's moving beyond healthcare.
It has that capability, and it will indeed in the future, although I have to say that our keen focus for the moment remains healthcare.
Now, Geoff, I noticed in the media recently you've been making some announcements about an acquisition. Can you share any of that with us?
Yes. In August, we announced because we are a publicly listed company, we need to adhere to continuous disclosure. And in August, we announced to the market that it was our intention to purchase a Belgian-based in vitro diagnostic company. So that is a Belgian-based company that makes pathology tests that they will sell to pathology companies. So in the Australian context, the company that we're looking to buy, DIAsource, would sell to the likes of Sonic. When your blood goes to Sonic, Sonic will buy a range of tests to perform on your blood, and those tests could come from Roche or Abbott or Beckman, or indeed DIAsource being the company that we're looking to acquire. The logic of the acquisition from our perspective is that we have an enabling technology that allows DIAsource to produce more sensitive tests or produce tests that cost less. So that's the logic for the deal. Yesterday, we announced to the market how we intended to fund that. And the acquisition, it's a 15.4 million euro acquisition in the first instance. And we've announced that we have signed a term sheet with a company that will provide a 10 million euro convertible note. And yesterday we announced that we would open an entitlement issue or a rights issue for shareholders to raise up to $12 million. So through a combination of both debt and equity, we'll raise the capital that will allow us to complete the acquisition of DIAsource. We expect that the rights issue will close on the 16th of December, and we would hope that the acquisition is completed before the end of this calendar year.
It sounds like a perfect match almost between the mix and go technology that you have and a provider of like DIAsource who's providing those diagnostic tools.
Well, that's why we entered into it in the first instance. But as we've immersed ourselves further into the due diligence, we've found that there are a variety of other benefits to both parties, in fact, that make it even more sensible. So, for instance, our technology can allow for better or less expensive immunoassays to be produced. But we've been looking around the world to find people that could do some very specific manufacturing on our behalf. And DIAsource is operating at about 60% capacity in its manufacturing plant, and they will be able to undertake that manufacturing on our behalf. In addition, they're growing very rapidly in the Asian region, and we're well placed to support their efforts in that region just because of our geography. Similarly, many of our most advanced projects are on foot in Europe. Yes. And for us to have a foothold in Europe will certainly improve outcomes from our perspective. So there are a variety of additional things that I guess we hadn't anticipated when we first indicated our interest in DIAsource.
Now, Geoff, a lot of our listeners, in fact about 95% of them, are clinicians of one kind or another, mainly in acute settings in hospitals or in aged care. And if you imagine them standing around the nurse's station at the end of shift writing up notes and charts and having a listen to the radio as they're doing it, what's the takeaway message for them today?
Well, I guess what we have is an ability to develop or assist in the development of more sensitive immunoassays. And for many of those clinicians that are your listeners, they'd be familiar with troponin, for instance, and the desire to have a more sensitive troponin test for the early detection of heart attack. Well, our technology allows that. And we believe, and we're dealing with many of the large players in the market now with a view to improving outcomes with the likes of troponin. Now, I use that as an example. There are many other assays that could benefit from greater sensitivity. There are many very, what might be considered to be important biomarkers that are perhaps fragile. The antibodies don't lend themselves to inclusion in immunoassays. Given this general avidity binding that we have the potential to develop assays using some of these more fragile biomarkers. So the message to your listeners is that we have an ability to produce very sensitive tests, and perhaps in addition, the costs could be reduced as a result of the inclusion of as much as 90% less antibody. And I guess from your users' perspective, they would want to know that the tests there's no issue in relation to stability or any other performance issues. And of course, that is not a concern, because before any of these tests hit the market, they need to go through the normal regulatory process. So whilst it is a different process, it must, before it hits the market, be demonstrated to be stable and if you like, the equivalent or better of anything that's on the market at the moment.
Yes, and I didn't ask the question about whether you'd been through those certification processes, so I just made the assumption that you had.
No, it in fact won't be us that does it. It'll be the manufacturers of the tests that would be responsible for those regulatory approvals. However, if we are incorporating Mix and Go, which is the name of the glue, into tests that are manufactured by DIAsource, we would be going through those regulatory processes in our own right.
Now, Geoff, my favourite question in every interview is about misconceptions, and in everyone's life and every aspect of work that we do, there are misconceptions amongst, in your case, I guess, scientists and clients and customers and maybe even patients. What's the biggest misconception about Anteo that drives you nuts and keeps you awake at night?
I guess there are two. When we first started out, we'd send out our glue to people that were developing immunoassays, and it would often work and often work very well, but sometimes it wouldn't. And generally, the reason for that failure was that the people that were doing the development didn't follow our instructions. As a result of that, we've now started to produce kits, and through selling kits, we find that we more routinely get the outcomes that we're looking for. So number one is follow the instructions, and finding new and better ways to get people to follow the instructions. That would be one of the things that keeps me awake at night. And in addition, because it is a different approach to binding, as I said, the most conventional approaches would be either passive binding or covalent. There's a concern that there might not be the same level of stability as with using coordination chemistry as exists for covalent. And I guess we've shown many times that it is an extremely stable approach to binding. And in fact, because it's avidity, it's a bit like Velcro. Any one of the bonds is very weak, and they break and they reform. And when you sit in a comfortable chair, it molds around the folds and curves of your backside. Well, that's a bit the way our glue binds to antibodies. Now, antibodies are fragile biomolecules that can be damaged very easily. This avidity binding process actually lends itself well, but because it's new, and of course because potentially lives are at stake, there's a degree of caution and reluctance to adopt a new technology. And so what keeps me awake at night is trying to find ways, undertake tests to demonstrate conclusively to any naysayers that there need be no concerns. And I think that those concerns of mine will be addressed as more and more people adopt it and it becomes general practice.
Well, Geoff, I can't help with the first concern you have. Reading the manual is something I'm afraid you'll just never get men to do. You'll only manage that if you manage to get all women working in the testing environment, because men have just, for thousands of years, not read the instructions. As for the other one, we can probably help a little bit with that. Some of our audience today listening, with a little bit of luck, will have heard you speak and will be saying, oh, that's interesting, I'd love to find out more. That leads me nicely to the website, The website is anteotech.com. That's spelled ANTEOTECH.com. That's also available on our website at the transcript if you missed it here. And that's where you can get in touch with my guest today and find out all about the Mix and Go glue that we've been discussing. Now, Geoff Cumming, thank you for your time. Geoff's the Managing Director and CEO of Anteo Technologies. I do appreciate you making yourself available today, Geoff.
Wayne, it's been a pleasure, and I'll be happy to field any questions that might be prompted by what I've had to say.
Thank you very much for that. If you've just missed the conversation I've been having with Geoff Cumming, the good news is there is a transcript on our website at www.hpr.fm and also links to an audio archive of the interview, which is on both YouTube and SoundCloud. You've been listening to Health Professional Radio. My name is Wayne Bucklar.